Hi everyone, welcome to the next instalment of the seminar series for students this year. Thanks for coming. Today it's our great pleasure to hear from Dr. Tracy Putoczki. She is the most recently appointed laboratory head in the inflammation division here at the Walter and Eliza Hall Institute where I work, and even in the short period of time that she's been in our division, we've already started a number of collaborations owing to her exceptional collaborative interests, and they specifically centre around the gastrointestinal tract, and Tracy has really established some of the best and only facilities in Australia for imaging this in some particularly unique ways. And specifically her research sort of revolves around the analysis of cytokine signalling, in particular gp130 signalling, with emphasis on the cytokine interleukin 11. And that's particularly relevant and with links to the industry, CSL and other pharmaceutical companies as well, in the treatments of some of these diseases like Crohn's and other things that she'll tell you about today, hopefully. So thank you very much, Tracy.
Thanks, Seth. So today I'm going to attempt to introduce you to two of my favourite topics, the first being inflammation-associated cancers, and the second being gp130 signalling. Now, these are really two distinct areas of research, and hopefully by the end of my talk, you'll start to see that they really do have quite a unique relationship. Now, before I introduce you to an inflammation-associated cancer, I really want to take a moment just to focus on the word cancer. This is a word that we use in my lab on a daily basis when we're talking about the samples that we're using or the grants that we're writing. But when we walk out the door of this building and in our personal lives, if we hear this word, it's actually one of the scariest words that you'll ever hear in your personal life. And that's because here in Australia, we expect that half of the Australian population will be diagnosed with cancer in their lifetime. So half of this room, half of our friends, half of our family members, half of our colleagues. In 2014, it's predicted that about 130,000 Australians will be diagnosed with cancer. This is any type of cancer. And there's a higher mortality rate for those with poor socioeconomic status. So your ability to access the type of medical care that you need really impacts on your survival. And for all cancers in Australia, the survival rate's about 66%. So that means 5 years after your initial diagnosis, only about 66% of those patients continue on. So as young scientists, there's a few things that we can do here. We can learn how cancers form and better understand how cancers form, and we can work towards developing better treatment options. And these are the two sort of underlying themes I'd like to address today. So there are a number of ways that we can understand how a cancer forms, and you've been introduced to these over the last few seminars. Just last Wednesday, Chris Burns introduced you to the NCI 60 panel of cell lines. So cancer cell lines were isolated in the 50s, so quite a long time ago. The first cell line isolated were HeLa cells, which many labs right now will have out in their incubators, and these were isolated from a cancer patient with cervical cancer. Now, one of the reasons we like to use cell lines is because they've been around for a really long time. That means that we understand quite a bit about them. We know how they grow. We know what proteins they make. We know what genes are mutated within them. So one of the things that we often do is downregulate or upregulate genes within our cell lines using a range of techniques that are quite simple to do in the lab now. Or when we develop drugs, we treat the cell lines and have a look at when their IC50 is, or so the rate of 50% of the cells dying. Now, because we understand so much about them and how to treat them, we've also managed to adapt them so that they'll grow in immunocompromised mice as xenografts. Then again, we can take our favorite treatments and see how they affect a tumor forming in vivo. But one of the things I want you to take into consideration is this really the full picture, and is this the best way for us to handle cancers? It's most definitely the way that most labs approach cancers. But if we have a look at cell lines, and this is an image of DLD1 colorectal cancer cells. So they were discovered by a guy with the initials DLD. He discovered 4 of them. This is the first of them. These were isolated in the 70s. They're my favorite cell line to work with because they have a lot of mutations. So as far as cancers go, they're pretty bad. And you can have a look at the cells here growing in a monolayer, and you can see them invading into the areas where there's no cells. Now, this is a human colorectal cancer tumor. And even to the untrained eye, you can see straight away there's a few different things. Here we have our epithelial cells. Some of these are mucus-producing cells. We have immune cells, and we have fibroblasts. So if you don't remember anything else from the seminar today, the one take-home message I want you to take in is that tumor cells don't live in isolation. We have to take things beyond just the petri dish and start considering what else is around the tumor cells. So we know that tumors have mutations, and this is just an image of beta-catenin staining in brown. When we see nuclear beta-catenin staining, that indicates to us that there's a mutation somewhere within the Wnt signaling pathway. We know that there are immune cells around tumors, indicated here by CD3 staining. But these aren't things that we've just realized. These things were first observed by Rudolf Virchow in 1863. Now, he's what we consider to be the pioneer in inflammation-associated cancers, and he hypothesized that cancers initiate at the sites of inflammation. It wasn't until 2004 that Time magazine did a special on inflammation-associated cancers, right around the time where some of the biggest heavy-hitting papers started coming out. And this is really when we started realizing that the inflammatory microenvironment around a tumor had quite a significant role in how a cancer progressed. So it took us 150 years to get to this realization. So what is an inflammation-associated cancer? To my mind, almost every cancer is associated with inflammation in some way or another. These cancers are age-related diseases. So this is my artistic impression of a baby growing out to adulthood. We tend to get the cancers late in life. There's a couple of ways that we think about how we get these cancers. Either we inherit a mutation. Now, it's not often that babies have cancer straight away. It takes a few things for that cancer to initiate. So perhaps there are a number of insults that we experience over life, and then the cancer initiates. Another way is that perhaps we have a number of insults throughout life. This results in mutations in cells. Once these cells have mutations, these insults then allow the cancer to grow. So for the purpose of the data that I'm going to present to you today, we're going to define a chronic inflammatory cancer as one where chronic inflammation, so these insults, promotes the onset and progression of cancer. And there's a second take-home message that I want you to remember today, is that regardless of how we get to the cancer, the tissue microenvironment can determine whether or not a cancer cell will progress, stay dormant, or whether or not it can be destroyed by our immune system. So in order to demonstrate to you how cytokines can be involved in inflammation-associated cancers, I'm going to introduce you to 4 different types of inflammation-associated cancers. They're probably classically the ones that we most often work on in the research lab, and they're probably the most common ones within the population. So the first is hepatocellular carcinoma. Within Australia, the risk is relatively low, considering compared to some of the other cancers. So 1 in 139 people will be diagnosed with this cancer. The 5-year survival rate, though, is not very good. 16% of these patients will succumb to the disease within 5 years of being diagnosed. Now, in terms of how we think we get hepatocellular carcinomas, they're linked quite strongly with chronic hepatitis infections, cirrhosis of the liver, which can be caused by alcohol-induced damage, and also obesity, which is a new disease that we're considering to be part of the inflammation-associated diseases. The second is pancreatic cancer. This cancer is a bit more common in Australia, with 1 in 73 people diagnosed, but the 5-year survival rate is abysmal. As far as cancers go, this is the worst prognosis that you can have with pancreatic cancers. Most of the treatment options that are available are really only palliative. We don't really have a treatment option to cure this cancer. We know that you can get pancreatic cancers from inherited mutations. We also now know that chronic pancreatitis and diabetes can contribute to the risk of developing this cancer. The third cancer I'll introduce you to is gastric cancer. The risk in Australia is 1 in 86, with a 5-year survival of 26%. We know that gastric cancers can arise from inherited mutations, but more commonly we associate them with Helicobacter pylori infections and high-salt diet. And the fourth cancer that I'm going to introduce you to is colorectal cancers. This is one of the highest incidences of cancer within Australia, with 1 in 46 being diagnosed with this disease. The 5-year survival rate is 66%. Now, one of the reasons we have a better survival rate here is because Australia has been very good at implementing screening programs. If we can catch this cancer early, we have many treatment options for these patients. Colorectal cancers can arise from inherited mutations, but it can also arise from chronic inflammatory bowel diseases. So how exactly can an inflammation promote cancer? I've introduced to you the tumor microenvironment that we see around a cancer. And this tumor microenvironment can produce a number of cytokines. These are soluble molecules that allow cells to communicate with each other. So while the tumor microenvironment can make cytokines, these cytokines can signal to cancer cells. These cancer cells can then signal back to the tumor microenvironment. So we have a series of autocrine and paracrine interactions happening. So cytokine comes from the Greek words meaning cell movement. They really serve as message molecules between different cells, and they were first discovered in the 50s, so we've known about them for quite a long time. There are over 150 different types of cytokines. Within those, there are greater than 40 interleukins, and interleukins were named because of their crosstalk between different leukocytes, and they have a broad range of biological functions that we don't fully understand. So I've been asked to talk to you about the gp130 cytokines, and in order to do that, I need to introduce you first to gp130. So gp130 is a beta subunit receptor. It has a series of domains, including a cytokine binding module and fibronectin domains in the extracellular portion of gp130. It was first identified in the late 70s. One of the important things about this receptor is that it has no cytokine bonding capability by itself. It requires other receptors in order to initiate cytokine signaling. And it has no intrinsic tyrosine kinase domain, so it needs a complex to form in order for JAKs to come in, which then phosphorylate its cytoplasmic tail. So the initiation of signaling requires a number of complex events. The crystal structure for the extracellular domain of gp130 was solved here within WEHI. Yubin and Nadia solved each of these regions, so you can see the Ig-like domain, the cytokine binding module, which is an elbow, and the 3 fibronectin domains. So a lot of the history for cytokines and a lot of the cytokines I'm going to introduce to you today actually happened within the walls of this very building. So how can gp130 be involved in cancers? gp130 is expressed ubiquitously, so it's on all of our cells. So for the purpose of my talk today, we're going to focus on a cancer cell. In order for a signal to happen, we need a cytokine. We need a cytokine-specific receptor. And then a number of downstream events can occur for signaling to happen. Now, Jeff very nicely introduced this to you as the simple series of events that happen in a footy game. So we have a kinase that phosphorylates gp130 and allows for phosphorylation of the stats. Now, I grew up in Canada, so I didn't really understand this analogy. So I see it a little bit more like this. We have our activated stats that are phosphorylated that are trying to get into the nucleus to have a signal. And we have SOX back here, which is trying to put the signaling into check. Now, I'm married to a Kiwi, so I see this another way as well. We have our cytokine here, we have our receptors, and we have our JAKs, which help to get the signal going. Now, regardless of how you see this signaling pathway, the ultimate goal is to activate downstream genes, and that only happens when STAT3 gets into the nucleus and regulates the transcription of genes. So which cytokines use gp130 to signal? There are a bunch of them. My favorite two are IL-6 and IL-11. These two cytokines are unique because they utilize gp130 as a homodimer, and they're the only two cytokines that do this. So there's 2 gp130 molecules, 2 IL-6 receptors, and 2 cytokines, and they form a hexameric complex. Other members of this family utilize gp130 heterodimers. Many of these use the LIF receptor. This includes LIF, ciliary neurotropic factor, and cardiotrophin-1. Now, LIF has a really long history here within WEHI. It was discovered and cloned here, and there are a number of people within this building that can give you a much better history on LIF than I can. Other cytokines include oncostatin M, which uses its own receptor, IL-31, which doesn't actually use gp130, so I'm not sure how it got into this family. It uses oncostatin M receptor, and IL-27, which uses a series of complex receptors as well. So we group these into the IL-6 family. So often we refer to these as the IL-6 family of cytokines because of their shared use of gp130, ignoring IL-31 here, or the IL-12 family. And IL-27 is actually part of the IL-6 and the IL-12 family of cytokines. Now, one of the things that I find most interesting about these cytokines is these are really complex interactions, and gp130 is able to interact with all of these in different ways. And one of the fundamental biological significance of these is that when these cytokines find their specific receptors, they induce similar as well as different downstream responses. IL-6 and LIF are the most well characterized. As I mentioned, LIF's history is within this building here. But most of our understanding about how these cytokines signal is based on IL-6. So many of the examples I'll give you today will be focused on IL-6. So what do we know about the cytokines? If we look at their sequence homology, they're actually quite different. So this is just a table looking at each of the IL-6 family cytokines compared to IL-6, looking at their sequence identity and their sequence similarity. So they're all 20% or under, so they're not very similar. So how do we group these into the same family if they don't actually look very much alike? And in order to do that, we use some of their structural features. So these are 4-helix bundle cytokines. This is one of my favorite reviews when I talk about these cytokines, because you'll notice here are the crystal structures that were solved by 2009. Since then, IL-27 has been solved, but there's a great big hole here. And as many of you will know, this is one of my favorite cytokines. So we sought to fill the hole. It took us 4 years, but we've got the structure of this little baby cytokine now. So interleukin-11 does look very much like interleukin-6, but there's a few differences, which we're hoping to get past our second review and get published shortly. So we know they're 4-helix bundle cytokines. We know they're similar but different. What do we know about their receptors? So again, if we look at the sequences of the receptors for the IL-6 family cytokines compared to IL-6 receptor, they're not very similar. And in fact, LIF, oncostatin M, and WSX are quite a bit bigger than the other cytokines. If we take the same review and look at what we know about the structures of these cytokines, many of the receptors, the structures have been solved. This viral IL-6 doesn't require a receptor. It's the only cytokine we know of that can act directly with gp130. But again, there's a great big hole here for IL-11, and I don't really like having holes when we're trying to study what cytokines do. So we've been playing quite a bit with protein that we've expressed for either recombinant IL-11, its receptor, and looking at how the two form complexes and how they behave. This is just an analytical ultracentrifugation showing the complexes that these form. So it's a segmentation velocity plot. We've got the first crystals for IL-11 receptor and the receptor and complex. So you can see here we've got needle clusters, and here we have rod-like crystals. And we're getting diffractions around 2 angstroms, so we're not too far off solving the first crystal structures for these cytokines. And this is really important because one of the questions that my lab is trying to address is whether or not IL-11 is really just like its sibling IL-6. So how do the cytokines form complexes? We don't really fully understand how this is done. Everything that we do understand comes from models in IL-6. So here we have our IL-6 ligand and our IL-6 receptor alpha. There are 3 cytokine binding sites within our ligands. So the first binding site in IL-6 interacts with the D2 and D3 domain of IL-6 receptor. So I often see this as the bum of the cytokine. Then in the presence of gp130, IL-6 is able to interact through site 2 with gp130. So here we have a bum-to-bum formation. We have the D2, D3 regions of gp130 and IL-6 receptor alpha. Then the third binding site in IL-6 interacts with gp130 in the second trimer complex. So we get a juxtaposition of the cytokines and the formation of a hexameric complex that looks a little bit like a donut. Now this has been solved for IL-6 receptor. It's been solved for the LIF receptor, but it hasn't been solved yet for the IL-11 receptor, which is something that we're working on at the moment. So we still have quite a bit to learn about these cytokines. One of the complicating features is when they were all identified, they were called numerous different things. So when you look at old literature, you have to keep in mind gp130 can be called IL-6 signal transducing receptor, CD130, IL-6 receptor beta, and a whole series of things. But what I really want to point out is the disparity between what we know about these cytokines. As of last week, there are almost 85,000 publications in IL-6, compared to just a few thousand, not even in the thousands yet for the other cytokines. So we still have quite a bit to learn, especially because cytokines are really important for a number of different biological processes. So just to recap quickly, in case I've lost anyone along the way, tumors have a tumor microenvironment that is really important to the progression of cancers. They produce cytokines. The cytokines can act on cancer cells. This happens through interacting with gp130 and cytokine-specific receptors. This allows JAKs to phosphorylate gp130. This provides docking sites for STATs, which then become phosphorylated. They translocate to the nucleus and then start to interact with their favorite downstream target genes, and we get a number of genes that have a role in cancer. Now, I haven't gone into too much detail about these genes specifically, but we consider STAT3 to act amongst many of the different hallmarks of cancer. Now, the hallmarks of cancer were defined quite a few years ago as all the features that a cancer cell needs to survive. So STAT3 is able to induce genes that allow a cancer cell to continually proliferate, to evade growth suppressors, to undergo invasion and metastasis, to continually divide, to have a constant source of blood supply, and to not undergo cell death, which they should do if they were normal cells. And amongst the newest hallmarks of cancer is this idea of the ability of the cancer cell to avoid immune destruction and the ability of tumor-promoting inflammation to push the progression of a cancer. So if we want to define the role of cytokines within any disease, there's a few ways that we go about it. The first is generally to make knockout mice. So if we remove gp130, it's embryonic lethal. The mice don't survive. So that tells us gp130 is pretty important for many of the requirements of life. If we remove IL-6, the mice are viable and healthy, but if we start infecting them, we start to see that they have defects in how they respond to infection. If we remove IL-11 receptor, we know that the females are infertile, but otherwise the mice are viable and healthy. If we remove LIF receptor, the mice are neonatal lethal. Same for CNTF receptor. Oncostatin M receptor knockout mice have compromised hematopoiesis. And if we take out STAT3, which is downstream of all these cytokines, the mice are embryonic lethal, again telling us that STAT3 is essential for life. So knockout mice are quite useful to understand how cytokines contribute to disease, and I'm going to focus on three: the IL-6 knockout mice, the IL-11 receptor knockout mice, and the STAT3 knockout mice. And these are all mice we have here within WEHI. So as I take you through the different types of inflammation-associated cancers, I'm going to use a formula. I'm going to introduce you to the human disease and what we know about it in the context of cytokines. I'm going to introduce you to the animal models for that disease, and this is a really important way to get those tumors in the presence of a tumor microenvironment. I'm going to give you the experimental outcomes for some of the publications that I'm going to introduce, and I'm going to give you a little snippet of what we think it means. Now, I put these inside of a box because as scientists, it's really important that we don't forget to think outside of this box. And I'm going to finish with a couple little examples of experiments that I don't have an answer to, but things for you to think about. So the first type of cancer, hepatocellular carcinomas. I've told you the risk factors are hepatitis infections, cirrhosis, and obesity. Now, often we think of obesity as a body mass index of greater than 40. We know that people with a body mass index of greater than 25 have a high incidence of the onset of hepatocellular carcinomas. So I'm going to focus on a model of obesity because this is a new area of research. And because of its prevalence, the current thoughts in the field are that obesity is actually a stronger risk factor than hepatitis infection. So how do we get a liver cancer? So we have a chronic injury. Say this is inflammation caused by obesity or other factors. We have a bunch of immune cells recruited to that site, and they cause damage. This results in death of some of the cells within the liver and the promotion of angiogenesis so that the cells have a food source to survive. Now, this early stage of fibrosis can actually be resolved. So if these things are detected early, we do have ways of treating them. But if it's not, and as this progresses, we get disruption of the architecture of the liver and loss of function. As a result of that, we have an increased amount of cell death. And as a result of that, as the liver attempts to repair itself, it repairs itself abnormally. And we end up with a stage of cirrhosis. Again, this can be regressed back to fibrosis if caught earlier, but if it's not caught, then we get the development of hepatocellular carcinomas. So what do we know about liver cancer and cytokines? Well, very, very, very recently, we've started to identify genetic polymorphisms in IL-6 and IL-6 receptor that link to the susceptibility. These are polymorphisms that may alter how these cytokines interact at the protein level and the signaling downstream. We know that there are gain-of-function mutations linked to about 40% of inflammation-associated hepatocellular carcinomas. And we also know that IL-6 levels are elevated in obese patients. So if we have a look at what's available in the literature, you can see here we have our increased expression of IL-6 in hepatocellular carcinomas. This decreases after these tumors are removed. If we look in our gain-of-function gp130 mutant patients, here in the black bars, we can see that we have an increase in all of the things downstream of the IL-6 signaling pathway, so including hyperactivation of STAT3. And as a result of this, you can see the brown staining here, we have an increased amount in activated STAT3. Now, we often look at phosphorylated STAT3 as the active form of STAT3, and when we see nuclear staining, we assume it's in the nucleus and regulating downstream target genes. So overall, we consider that about 40% of hepatocellular carcinomas have elevated IL-6. So if we want to understand what IL-6 is doing in these diseases, we often turn to mouse models. In the first mouse model that I'll introduce to you, we refer to as the DEN model. Mice are injected with this carcinogen at 2 weeks of age, and then we can give them a high-fat diet and monitor tumor progression over time. Now, if we look at mice on a high-fat diet, you get a bit chubby. In mice on a low-fat diet, you can see the incidence of tumors just in a wild-type mouse is much greater in mice on a high-fat diet, when mice on a low-fat diet are either refractory or develop very few tumors in this model. And just to show you in these mice, on a low-fat diet compared to high-fat diet, we have an increased amount of IL-6 within the serum and an increased amount of IL-6 in the non-tumor liver compared to the tumorigenic liver. So what happens if we take IL-6 away from these mice? Mice that have no IL-6 so first of all, if we give the mice low-fat diet, and these high-fat diet, dark circles, open circles, mice that have no IL-6 don't quite gain as much weight as mice that have high IL-6. If we have a look at the tumor incidence, we can see in the high-fat diet, if there's no IL-6, there's a reduction in tumors compared to the mice on a low-fat diet. And this can just be quantified by the number of tumors, the size of the tumors, and the incidence of the tumors. Now, the take-home message from this publication was that macrophages are really important to the production of IL-6 in obesity as well as within liver tumors. If we look at wild-type mice on a low-fat diet to high-fat diet, we see an increase in influx of F480 positive macrophages. And in the absence of IL-6, we lose this influx of macrophages. So the loss of IL-6 can reduce overall tumor burden in a mouse model. So what do we think this means? People who are subject to obesity have an increased level of overall IL-6. The macrophages within the liver can produce IL-6, and so can the hepatocytes within the liver. Now, liver cells express IL-6 receptor, which is indicated here by the pink staining and this immunofluorescence. When they interact with the IL-6 receptor, and we have a promotion of liver inflammation DNA damage, we get activation of STAT3 within the liver cells, and this promotes downstream pro-survival phenotypes that can promote hepatocellular carcinomas. So from studies like this, one might start to assume that IL-6 might be a new therapeutic target for liver cancers. If we can generate reagents to inhibit signaling by IL-6, perhaps we can reduce the progression of liver cancers. Well, what about pancreatic cancers? So I've told you pancreatic cancers can be the result of inherited mutations, pancreatitis, or diabetes. And I'm going to focus on pancreatitis because this is one of the newest areas of research for pancreatic cancer. Now, I'm not going to introduce you chronic alcohol diseases, but I've just put this here for the Friday night turf crew. 80% of chronic pancreatitis is due to long-term alcohol abuse. So we understand the genetic predispositions in the genes associated with the onset of pancreatic cancer. So here we have your normal pancreas, and it develops through a series of histological grades that we use to define the stage of the pancreatic cancer. So changes in these cells are associated with mutations in different components of the pathway, and this leads to the development of the cancer. And we've only just realized that along this grade of pathological scoring, there's an increased amount of inflammation and desmoplasia, so the stromal reaction to the cancer. So we know that genetic polymorphisms exist in IL-6 in pancreatitis patients, but these aren't significantly linked to their progression to pancreatic cancer. We know that IL-6 is a marker of severity in pancreatitis, and actually the levels of IL-6 peak before the levels of C-reactive protein in pancreatitis, and this is a protein marker we often use for inflammatory diseases. We know that IL-6 is elevated in pancreatic cancer patients. But what nobody's ever looked at before is IL-11, which is, of course, what we're doing. And we've seen an increase in expression of IL-11, especially at these late stages in pancreatic cancer patients. And if you have a high amount of SOX3 expression, and SOX3 is a target for STAT3, So SOX3 can be associated with high levels of cytokine signaling. The higher your SOX3 expression, the poorer your prognosis. So there's a few models that I'm going to introduce you to for pancreatic cancer. The first my lab has here at WEHI, where we take a PDX mouse. So this is a Cree specific for your pancreas. We cross it to a mouse that has mutations in KRAS. When these are crossed, we get activating mutations in KRAS. The mice develop pancreatic tumors, and they have to be culled within about 6 months. Now, this is what that histopathological progression of pancreatic cancers looks like in these mice. Here we have your normal islets, ductal epithelium, and Asner cells. As they go on to PANN1, you can see we start to form cuboidal cells. In PANN1-2, we start to get production of mucus cells and loss of organization of the architecture of the cells. By PANN3, things become quite chaotic, and by the time we have the full-blown cancer, we have a complete stromal reaction to the cells. Now, the second model that I'm going to introduce you to is how we induce pancreatitis. So we inject the mice with a chemical. Wild-type mice develop pancreatitis. Mice that have this activating KRAS mutation develop earlier onset pancreatic cancer. And this was a really important experiment done by another group a couple of years ago, which was the first indication that inflammation could speed up the development of pancreatic cancers. Now, I haven't been completely truthful in how IL-6 family cytokines signal, and I've left out quite an important component. IL-6 receptor alpha has a membrane band formed, but this membrane band formed can also be cleaved, and we refer to this as trans signaling. So this soluble form of IL-6 receptor can interact with an IL-6 cytokine, and then it can interact with gp130 on any cell, so it bypasses the requirement for the membrane-bound form of this receptor. That means that any cell is now susceptible to IL-6 signaling. And now just to remind you about the bits that Jeff introduced you to. These JAKs then become activated. They phosphorylate the cytoplasmic tail of gp130 and provide docking sites for STAT3. These translocate to the nucleus, as I've mentioned, and we get genes induced that involve proliferation, survival, and angiogenesis. SOX3 is our target gene for STAT3 and also the negative physiological regulator of STAT3 activation, targeting the complex for proteasomal degradation. And just to contrast, interleukin-11 receptor is in a membrane-bound form as well. We haven't identified a soluble form of this receptor as of yet. So if we induce pancreatitis in a wild-type mouse, we get an induction in SOX3, and we get an increase in activation in STAT3. Now, this was a paper published a couple of years ago that caught my eye, because it's not very often that people look at IL-6 and IL-11 together. Now, they have a different Cree, but it's still a pancreas-specific Cree, and they have a KRAS mutant. And we see one day after induction of pancreatitis, an induction of IL-6, as well as an induction of IL-11. And this is dependent on STAT3. If we remove an allele of STAT3, we get a reduction in the IL-6 and IL-11 that's produced. Now, this wasn't something that was just a byproduct. If they took out pancreas cells and cultured them and stimulated them with the same chemical that we use to stimulate damage in the cells that induces pancreatitis, we get an induction of IL-6 and IL-11 that's dependent on STAT3. So this caught my eye. So we started doing a few things to see if perhaps IL-11 could do the same sorts of things as IL-6. And we started asking the question, why are both of these cytokines elevated if they do similar things and activate similar pathways? So this is the original paper's data. You see an elevation in IL-6 mRNA expression in the pancreas of these mice. Over time, it increases as the tumors start to develop. They see that IL-6 is expressed by macrophages within the pancreas. They see that if they treat cells isolated from these mice with IL-6 alone, they get an induction of STAT3. But with hyper-IL-6, which is a form of IL-6 fused to its receptor, they get a really strong induction of STAT3. So again, this bypasses the need for the membrane-bound receptor and induces full-blown STAT3 activation. So they've set the precedent that IL-6 is elevated in their mouse model of pancreatic cancer. So what happens if we remove IL-6? If we take out IL-6 as it is, we take out both classical and trans signaling because there's no longer a ligand to signal through either receptors. And if we take out, or if we express soluble gp130 as a transgenic, this is a form of gp130 that can mop up the soluble IL-6 and IL-6 receptor inhibit signaling through trans signaling. We see that we get a reduction in the progression through these different stages of pancreatic cancer. And you can see this as well within their histology. So here we have our bad cancer, and here we have pancreases still looking a little bit more like a pancreas. Now, they showed that in the absence of IL-6, or when soluble gp130 is expressed that inhibits trans signaling, they get a reduction in STAT3 activation. And these are the kinds of plots that I look at and say, well, that's nice, STAT3 is reduced, but there's still STAT3 there. What's producing the rest of the STAT3? What's IL-11 doing in this context, and is it contributing to the remainder of the STAT3 activation? So we've got the same cross of mice. This is work ongoing in our lab at the moment. If we weigh the pancreas, which is an indication of the number of tumors, because tumors weigh more than the pancreas, or the edema that happens in the pancreas as a result of the pancreatitis the mice are experiencing, you can see that this is the weight of a wild-type mouse. This is our KRAS mutant mouse. And if we remove just one allele of IL-11 receptor, we start to get a significant phenotype in these mice. Now, what about a third model? Because there's a number of mutations involved in pancreatic cancer. So this is the third model we have here at WEHI. This is our first mouse, which is now crossed to a mouse that's going to have mutated p53 and a YFP allele. And the reason we have the YFP allele is because this mouse actually metastasizes. So the YFP allows us to track where the cells are metastasizing. Now, if we cross this mouse to the IL-11 receptor knockout mouse, and these are a pain to genotype, we get mice that have a significant reduction in the progression of disease. This is only half of the alleles having IL-11, so we still have half the signaling. Also, without half of IL-11 receptor signaling, and I call these the PKTY mouse because it's the PDX, KRAS, TP53, YFP. It's too much to write below the sign. We get a significant reduction in STAT3 activation. So this tells us that IL-11 must have a role in pancreatic cancer as well. And so we'll be looking at understanding how IL-11 is involved in the onset of pancreatitis and the progression of pancreatic cancer in these mice. And hopefully we publish this before somebody else does. Now, just a quick overview of what we do understand about pancreatic cancer. We know that macrophages can secrete IL-6. We know that many cells within the tumor microenvironment have IL-6 receptor cleaved off of them to create the soluble IL-6 receptor form. We know that pancreas cells can secrete IL-6 and IL-11, and mutant KRAS has actually been shown to induce the transcription of these genes. This results in continued inflammation and survival. And the current thoughts in the field are that this precancerous set of lesions require trans signaling, and the progression of the actual cancer requires IL-6 signaling once the cell becomes self-sufficient in producing the cytokine. So we might start to take a few different conclusions from this type of data. Perhaps giving soluble gp130 is a new target to control the progression of this disease if it's diagnosed early. Perhaps IL-6 is a new therapeutic target for established disease if we need to control the growth of the actual tumor. And perhaps IL-11 might be a therapeutic target within this context as well. And then we need to start asking the question of which cytokine is the best of these to target. So moving on to gastric cancer. Gastric cancer also has a number of inherited mutations. Can also be induced by H. pylori infection, and this is what we most commonly associate gastric cancer progression. Within Australia, we predict that about 50% of the population will have a colony of Helicobacter pylori within their stomachs by 50 years of age. Now, this isn't necessarily a problem because this can colonize the stomach and be completely asymptomatic. But when it does colonize the stomach and cause symptoms, Helicobacter pylori, which is a gram-negative bacteria, can induce chronic gastritis. This long latency of chronic gastritis can induce changes within the stomach that lead to atrophic gastritis, intestinal metaplasia, where the stomach starts taking on features of the intestine, including mucus production, dysplasia, and the formation of cancers. So we know that IL-6 has a role within human patients. IL-6 can predict the survival outcome after surgical resection of a stomach cancer. We know that IL-6 and IL-11 are both markers of tumor aggressiveness within this disease. We know that STAT3 activation correlates with metastases of stomach cancers. And we've shown looking at individual tumors within a cancer, so each of these doublet bars is the expression of IL-11 or IL-6 within one tumor compared to the normal tissue. In most tumors, we see an elevation of these two cytokines. And as a result of that, we see activation of STAT3 within the tumors. Now I'm going to introduce you to a mouse model that Matthias actually made as a postdoc at the Ludwig Institute many years ago. This mouse has a phenylalanine knock-in substitution at this tyrosine 757. So we refer to this as the FF mouse when it's homozygote for this phenylalanine knock-in substitution. Now, as a result of this mutation, SOX3, which is our negative physiological regulator of STAT3 signaling, is no longer able to interact with gp130. And as a result, we get hyperactive STAT3 only in the presence of ligand. So these mice have a number of problems. They have a pan-systemic inflammatory phenotype, but in particular, we're going to focus on the stomach. So this is what a wild-type stomach of a mouse looks like. This side would be the lumen, this is the muscle, and this is the outer stomach. We can see the antrum, the body, and the squamous epithelium of the fundus. Now this is what the stomachs of the gp130FF mice look like. You can see these massive adenomas that form in the antrum, and we can see all the different immune cells throughout the stomach. So we consider this an inflammation-associated gastric cancer model. Now these tumors have a pan-proliferation response, a number of atrophying and branching glands, gastritis, dysplastic cells, intestinal metaplasia, which is this mucus-producing phenotype. Now we started looking at what could be driving all of this. And Brendan Jenkins, who was a postdoc with TACE many years ago, looked at the expression of gp130 receptors, as well as some of the gp130 ligands, including LIF. And what he saw was an increased elevation of IL-6 and IL-11 within these mice. Now, to cut a long story short, we know that IL-6 doesn't actually have a role in cancer progression in these mice, and that IL-11 has quite a significant role, despite the fact that both of these cytokines, when injected into the mice, can initiate STAT3 activation. So I want to take you a little bit downstream of the cytokines for the gastric cancer models. If this is our wild-type stomach, here we have our fundus, our body, our antrum. This is our gp130 stomach. Here we have our fundus, body, and antrum. You start to see, if we remove one allele of STAT3, we have a significant reduction in tumor burden. And if we age STAT3 heterozygote mice over time, we continually have that significant reduction in tumor burden. So STAT3 is controlling the continued growth of tumors within these mice. This gives us a basis for treating the mice with STAT3 antisense oligonucleotides. So this is one way of inhibiting STAT3. And you can see when we treat the mice with the STAT3 antisense oligonucleotides, there's a significant reduction in tumor burden. So what does that tell us about gastric cancers? If you have a continual insult, like an infection from Helicobacter pylori, you'll get production of cytokines like interleukin-11 from a number of different cell types within the stomach. These cytokines can then signal to the epithelial cells, which express interleukin-11 receptor and gp130 and can produce interleukin-11 themselves. We get activation of STAT3 and all of its pro-survival downstream genes, and the formation of gastric adenomas. So this tells us that STAT3 is also a potential therapeutic target for gastric cancer. So the final model that I'm going to introduce you to is colon cancers. We know that colon cancers have inherited mutations, and we know that they can arise from inflammatory bowel diseases. Now, there are two specific types of inflammatory bowel disease, ulcerative colitis and Crohn's disease. Both of these inflammatory bowel diseases are associated with an influx of inflammation and an increased activation of STAT3. Now, patients that have this increased influx of inflammatory cells and increased STAT3 may also have different mutations accumulate within them. Now, we know that genetic predisposition only contributes to 20% of colorectal cancer, so something else helps them along. And so the progression of a cancer goes from normal epithelium to aberrant crypt foci through to a carcinoma that can metastasize. This is associated with a number of different mutations at different stages. We now appreciate that cytokines have a really important role in promoting the growth of these tumors. So we know that IL-6 is elevated in colon cancer patients. We know that IL-11 is elevated in colon cancer tumors. And we know that inhibition of STAT3 improves chemotherapy outcome. Now, this is something really important to consider. Since these cytokines have so many pro-survival activities, and since we know so many therapies become refractory over time, is it these STAT3 cytokines that are promoting the survival of the cells that are refractory? And if we inhibit STAT3 at the same time as our standard of care, could we improve the outcome of these therapies? Now, similar to what I've shown you for gastric cancer, if we look at IL-11 and IL-6 within the same tumors of patients compared to normal samples, we see that these cytokines are elevated in most tumor samples. And again, this is associated with elevated STAT3 activation. So I'm going to introduce you to the AOM-DSS model. This is how we model colorectal cancer, and we do this quite often here in WEHI. We inject the mice with a mutagen, and we give them 3 cycles of dextran sulfate sodium in the drinking water. Now, this is a luminal irritant that tickles the epithelial cells with the colon so that they start undergoing a wound healing response. And after 3 doses of this, we start getting formation of tumors. Now, we know in this model that in the serum, just after DSS, there's an elevation in IL-6. We know that IL-6 increases within the tumors themselves over time in this model, so by day 49 of this model. We know that these tumors also have elevated IL-11 and elevated STAT3. So this mouse model of colitis-associated cancer has both elevated IL-6 and IL-11. So which ones are important? Now, we published later last year comparing the IL-6 knockout mouse and IL-11 receptor knockout mouse side by side. You can see we get a reduction in tumor burden in the IL-6 knockout mice, whether we monitor them by endoscopy over time. We'll look at their total tumor burden at the end. And in the absence of IL-11 receptor, we get quite a shocking reduction in tumor burden in these mice. So this suggests that in this model, perhaps IL-11 has a more potent role than IL-6 in the progression of the tumors. So maybe the role of these 2 cytokines isn't quite the same. And given that we've based everything on what we understand about IL-6, perhaps it's important to start revisiting these cytokines. So how can we inhibit cytokine signaling? The ultimate goal of our work is to generate new therapeutics. If we know that there's an inflammatory microenvironment, we know that cytokine signaling happens. We can either take out the cytokine, we can take out the cytokine receptor, we could take out gp130, we could take out JAK, or we could take out the STATs. So there are a number of different ways that we can do this, and we need to work out which one of these is the best approach. And so using this CAC model, people have shown that if we give the mice soluble gp130 Fc from before when the tumors form, we get a reduction in the onset and progression of tumors. If we give the mice a neutralizing antibody against the IL-6 receptor from before when the tumors are formed, we get a reduction in the onset of tumors. If we give the mice IL-11 mutein, which is a peptide antagonist that inhibits the receptor from signaling, when tumors are formed, we get a cytostatic effect on the tumor, and they don't continue to grow compared to mice that don't have this. So we get a reduction in tumor burden in the presence of things that inhibit IL-6, as well as in the presence of things that inhibit IL-11. Now, we can also inhibit JAKs in this model, and we get a reduction in tumor burden when tumors are formed when we treat the mice with a JAK inhibitor. And again, we can quantify this difference. So it seems all these different ways will inhibit tumor formation. So some of the things that we need to start addressing is the side effects that these therapies may have as we start to make decisions about which might be best. So just to go over colorectal cancers, we know that they can be driven by colitis. We know that the length and duration of the inflammation leads to a more enhanced or worse disease. We know that cytokines like IL-6 and IL-11 can signal to the mutated cancer cell. We also now know that hematopoietic cells and fibroblasts are probably the predominant source of IL-11 within colorectal cancers. We know that if we inhibit a number of different parts of this pathway in colorectal cancers, we get a favorable disease outcome, at least in mice. So just to summarize the different ideas that I've presented to you today, gp130 cytokines are a unique family. There's a number of them. They have very specific ligand-receptor interactions, but redundant functions. And it's quite interesting to think about why we might have evolved to have so many different cytokines that can do similar things. gp130 itself is really interesting as a beta subunit receptor because it's polyspecific. How did it get to recognize so many specific cytokines in specific ways? And of course, one of the things we're starting to address is whether or not when these receptors come together, there's a difference in the quantity of signaling, the downstream activation of STAT3 in response to them. I've tried to hammer home that the tumor microenvironment is really important for the production of cytokines, and that these cytokines are really important in how cancer cells behave. So again, the tissue microenvironment really plays a strong role in determining the outcome of this cancer cells. We still need to understand from a full physiological point of view which cells make and which cells respond to different cytokines, and understand if there's specific diseases where one cytokine might be more important than another. And then to take this idea of combination therapies, using our cytokine inhibitors with standard of care treatments to see if we can improve therapy outcome. So what's in the clinic for the work that we've done so far? There are a number of STAT3 inhibitors, JAK inhibitors, and soluble gp130s that have gone into clinical trials. The JAK inhibitors have been terminated so far, unfortunately. For pancreatic cancer, there's a JAK inhibitor by Novartis that's about to go into clinical trials. For gastric cancer, again, there was a JAK inhibitor but this was terminated. And for colorectal cancers, there's nothing at the moment going into clinical trials. So there's quite a bit of room for improvement given what we know in animal models about the role of cytokines in these diseases. So I said I quickly wanted to leave you with something outside of the box to think about. Now, I've talked about inflammation, but I really haven't talked about the underlying triggers for inflammation. And we're quite interested in how our mucosal barriers, which include a mucus layer, regulate our host response to damage. And one of the things that I think is quite interesting is that when we look at a tumor, we see quite a significant change in mucus production. So mucus is this pink stuff here. And I wonder, perhaps the tumor is taking advantage of the fact that we've just gotten rid of one of its mucosal barrier protection modes. So bacteria can then get into the tumor here and trigger our host immune response. And this is one of the ways that the tumor can hijack our immune system to get those cytokines produced that help it to grow. So one of the ways that we're going to start looking at this, and this is really just a plug for a new piece of equipment that we'll be getting in our lab. This is our classic light endoscopy unit that CSL purchased for the Ernst lab in mine. And this is the new confocal endoscopy unit that we'll be getting that's been funded by ANZ. This is a 488 laser, so it allows us to track reporter mice so that we can look at different immune cells or cytokine-producing cells in vivo and track which cells are making them, when they're making them, where they're making them, and perhaps start asking why. Now, I have one quick plug to do. WEHI is putting a lot of time and energy into our students. We want you to have the best possible career development. And so there will be a next generation of student symposium. This is going to be an opportunity for the students to showcase what they're bringing to the table so that we know what our next generation of scientists can do. This is also an opportunity for the students to learn how to present their work within a forum, much more of a conference-style forum. So the abstract submission deadline will be in July. We'll have the information up on Catalyst shortly. And if you have any questions, please email the Student Symposium email. But once Catalyst is up, then you can look at the information. Now, just quickly, everything that we do in science, we do as a team. If anybody tells you otherwise, they're completely lying. I've just listed a few of the different people who have helped with the different aspects of the projects I've shown you. I've not done anything with liver cancer myself. Everything I've shown you is other people's published data. And of course, without our funding, we can't do anything at all. And I thank you for your attention.
Thanks very much, Tracy. Wonderful overview of gp130. And so do we have some questions? Perhaps I can just get started by asking you a sort of broader question about, you've provided some really compelling evidence for pro-cancer roles for inflammation, but one of the major things that inflammation does is to educate your adaptive immune response, and you mentioned also that this could potentially be a way to educate and to help fight oncogenesis. So is this a double-edged sword? Does inflammation also help prevent cancer by educating immune responses against disease?
So I absolutely think it's a double-edged sword, and I think one of the reasons we have so many different cytokines is the pressure and evolution of our host response to disease to have those cytokines. So my personal take is that IL-6 is our first response cytokine. It's helping all of the pro and anti-inflammatory cells to get into the tumor, fight the tumor infection, whereas perhaps IL-11 evolved to fix the wounding that's happening, the damage that's happened, and the tumors have hijacked that response a bit more specifically than IL-6.
Students, any questions?
Come on, I don't bite.
You showed that IL-6 was in the serum. Is it possible to use that as a diagnostic method of pancreatic cancer?
Yeah, absolutely. So those are the types of studies that people are trying to do. If we can detect why someone has a cancer before they have it, then that's fantastic. The trouble, as Seth mentioned, with things like IL-6 is they do come up when a number of different things happen. So if you had a cold, your IL-6 levels would be high. How do you separate someone who has a cold from someone who has pancreatic cancer, as an example? But those are the types of things people try to do.
How different are the STAT3 dependent transcriptional responses of IL-6 and IL-11?
Yes.
Are they activating very similar things?
They're nearly identical apart from a handful of genes. So there's a handful of genes that seem to be quite specific in the context of IL-6 in immune cells, in the context of IL-11 in epithelial cells. So that's one of the reasons why I say I think the two cytokines have diverged for different functions within the cell that the tumor is taking advantage of.
You mentioned, showed some really clear differences between IL-6 and IL-11. do you think the major difference is trans-signaling, or do you think it's without knowing the structures, it's hard to comment on the affinity of these various receptor ligand complexes or the downstream pathways that Ed's referring to?
So I personally think there's a few differences, trans-signaling being one of them. We haven't seen whether or not there's trans-signaling for IL-11 yet, so we don't know if that soluble gp130 is mopping up both at the same time. I think the other major difference is where the receptors are expressed. If you have more of IL-6 receptor or more of IL-11 receptor, you're going to be prone to having signaling by one of those two cytokines.
So you said that there's various drugs in the market when they use for IL-6. So does it affect the levels of IL-1 in the tumors as well?
Yeah, so if you start taking out one cytokine, you absolutely start taking out the other cytokines. Part of the reason for that is that they compensate for each other quite a bit in what they're doing. What we actually see in the mouse models is when we take away a cytokine over a prolonged period of time and then we stop that treatment, a lot of those inhibitors aren't inhibiting the transcription of the cytokine, they're just blocking them within the serum. So you take that away, you have this pool of things that are sitting there, then all of a sudden you get this rush of STAT3 activation, and then things start to become worse. So in our opinion, you have to be on a cytokine blocker for the duration of your treatment until the cancer is gone.
I'm just wondering, you focused on STAT3 as your downstream mechanism for IL-11 and IL-6, but they can also signal via PI3 kinase and MAP kinase.
Absolutely.
Have you looked at those?
Absolutely. No, absolutely. So in one of our publications last year, we showed that IL-11 could activate the mTOR pathway, and if you inhibit IL-11, you take out that pathway as well. For simplicity, I left out all of those other pathways, but they do absolutely do things. From my personal point of view, if we can take out something at the top and take out multiple pathways, that's much more effective than trying to take out one or the other.
We've got another talking at 1 o'clock, so well, one last quick question, then we'll...
Are there different levels of the soluble IL-6 receptor in cancer patients as opposed to healthy people?
That's a very good question, actually, and it's not one I know the answer to off the top of my head.
All right, then join me.